актуални въпроси, най-вече за лечението на главоболието и в първия случай за мигрената. И също така един доклад, който е посветен на лечението на двигателните нарушения. Но първият доклад, който ще представи професор Шарма, е посветен на едно изключително авангардно направление, а именно приложението на наномедицината и на нанотехнологията в наномедицината с оглед евентуално подобряване на диагностиката, лечението и превенцията на голяма част от неврологичните заболявания. И аз бих помолил професор Шарма да представи своя доклад. Please, Professor Sharma, I would like to ask you to present your speech.
Thank you, chairpersons and friends. First of all, this is my great honor to be here in my maiden visit in Bulgaria, and I feel very honored to share some of our results with you today. We are very much delighted to be in this great country. Before I go, I have to say that whatever I am telling you, this is not my personal view. Of course, this is not the organizational view or the US government or different organizations with I am working. So all these to be considered the view of our group. And here in the neurology meeting, I don't need to tell much about the blood-brain barrier because all of you are very much aware. But I can only tell that in the brain, the neurons are about 10 billion, but glial cells are much more than that, and even the endothelial cells even more. So we cannot ignore the role of endothelial cells in central nervous system diseases. This is our institute in India where I did my PhD in 1982, and I am a basic scientist. I did my doctoral work on blood-brain barrier in neuroscience in 1982. After that, we continue on this work, and this book we have edited from Elsevier in 2003. I can tell you that we consider blood-brain barrier as a gateway to neurological diseases because there are no other diseases in the brain where this barrier is not disrupted to large molecules such as proteins. But the good point is that they have receptors of drugs, and we can influence this barrier permeability by different kinds of drugs. As you can see here that this is one cerebral capillary and this is extracellular space where glial cells are lying. When this is leaky, it can influence not only the neurons but also glial cells. So in order to have healthy brain, we need to keep this barrier intact. One of the earliest results by Brightman in 1969 from NIH showed that lanthanum is 20 angstrom in diameter is stopped at the tight junction, and this was the first anatomical evidence that blood-brain barrier is maintained by the tight junctions between the endothelial cells. Then I worked with Professor Sharma in Germany on Humboldt Foundation Fellowship, where we studied changes in ultrastructural blood-brain barrier under hyperthermia. So here is our model. This is a rat spinal cord, and we injected lanthanum, and you can see that in healthy situation it is not leaking, whereas the basal lamina is intact. One neuron can be seen here, and this is the glial cell. We use this model to study blood-brain barrier under different conditions. But now we are also examining the barrier between brain and blood, because you know when any tracer substances is administered here, they are stopped at the luminal side of the barrier. But if some chemicals are injected in the brain, they are also equally stopped in the abluminal side of the barrier. It means that in healthy condition, both barriers are intact. But in disease situations, it appears that the brain-blood barrier is also broken down. But not very much research is done in this area. perhaps the brain-blood barrier is important because when many neurochemicals are released, then if that barrier is broken, it can come to the systemic circulation and will have rebound effects. Our hypothesis is that any kind of CNS insults, be it a stroke, Alzheimer, trauma, this blood-CNS barrier is open and produces many changes, cellular changes, and all could lead to cell death. We are working to prove or disprove this hypothesis so far. Now the question of nanoparticles. In last 10 years, we are very much aware about the nanoparticles and its existence in the environment. For example, if there is a polluted environment, there are many microfine particles are in the area that we breathe almost daily. Think about some of our soldiers who are posted in the Middle East, where they are exposed to sand or silica particles. They are doing every day and breathing this. When they get injured, probably their situation is entirely different than the injury can occur in a cleaner environment. So we summarize some of these aspects in a book. We also have a journal called Journal of Nanoneuroscience by American Scientific Publishers, and its editorial board is quite good. Then nowadays, this nanoparticles are the functions of also the government interaction. This is one of the Indian president, the sitting president. He wanted to establish a consortium between different countries to have this nanomedicine project funded. And also I have the pleasure that Andrew Shelley, who was Nobel Prize winner in 1977 in physiology and medicine, is also supporting this idea, and we are collaborating with him. And also another Nobel laureate, Aaron Shikanover, he is also very keen to study ubiquitin expression, on which he got the Nobel Prize, how nanoparticles can affect this. So in a nutshell, we are trying to understand the influence of nanoparticles on the brain, and on the other hand, we also want to give our drugs using nanotechnology to have better effect of those compounds. I am basically a pathologist, work with Professor Ingve Olsen in Uppsala University, and we feel that this blood-brain barrier is playing dominant role in all kinds of diseases. But at the moment, we cannot forget the role of nanoparticles, because they can produce neurotoxicity, but as well as nanodrug delivery can have better nanoneuroprotection. We are working with a lot of people, even from India. We have India-EU research cooperation on nanoparticles. But consider the situation of military, because our research is supported by US Air Force Research Department and also European Aerospace Research and Development in London, UK. This is a branch of American Air Force Research Laboratory, Swedish Strategic Research Foundation, and all other organizations. So you can see that these soldiers are exposed daily to silica dust. In some of the countries, most of the soldiers are protecting the borders like this. So they have a kind of a stress, a partial immobilization stress. So when they have silica exposure, they have a stress, their brain function is entirely different, and therefore we are collaborating with the director of US FDA at Jefferson, National Center of Toxicological Research, Dr. William Slicker, and we work with them. So first I will show you, now the time has come in neurology or any discipline that we should consider multiple factors in diseases, not only one factor. I can give you an example. If a stroke patient comes, he has many other side diseases like hypertension or diabetes. If we would like to treat with only a drug without knowing the comorbidity factors, probably the results are not good. And this could be one of the reasons that we have not so much success in clinical trials of even good drugs. So now we are considering different parameters. Even stress, how can stress influence the disease? How can hypertension or diabetes or even combination? So I will show you some observations on hypertension. We have this field stress model, and this is a chamber where we have 38 degrees Celsius, and the humidity and airflow is kept constant, and this is a simple model where we can have animals partially immobilized. We have measured glucose changes at different conditions. The rats were made hypertensive by tube kidney one clip method, and they were exposed to 21 means room temperature or 33, and they were also combined with immobilization. The point is that the maximum changes occur when these hypertensive rats with immobilization stress were exposed to 33 degrees Celsius. I will show you some example. The blood pressure after heat exposure came down, but the point is that we have also studied some cognitive functions, and this is a rotor experiment when normal rats can stay up to 60 seconds, but when disease rats will fall down. And this is also an inclined plane. Normal rats can stay here, but disease rats we have to lower down the angle. And on some of this kind of work in 2005, US FDA has given us some certificates. So we go ahead. The point is here that this is rotor performance. These are the normal animals. And I would like to draw your kind attention to see here that when the hypertensive rats with stress were exposed to 33 degrees Celsius, they were worse performer. Whereas the other conditions also here that these hypertensive rats immobilization 33 degrees Celsius, they also have the worst situation here. So the point is that when we have stress, when we have exposure to nanoparticles, then the situation is going to be worse here. This is also inclined plane angle, and these stressed animals with hypertension and silica exposure, they are the worst here. So the point is that nanoparticle intoxication after stress is more dangerous when they are exposed to high ambient temperature. This is an example of blood-brain barrier leakage, and you should look on these bars. They are the stress, hypertension, immobilization, and high ambient temperature. And the other were also without stress, nanoparticle exposure, and high ambient temperature. So the point is that different circumstances can make brain vulnerable. This is also example of brain edema, and as you can see here, that immobilization, hypertension, and silica exposure is the worst in all the cases. Neuronal injury, you can see here, the same situation occur, that these bars in different brain regions, cortex, hippocampus, striatum, thalamus, hypothalamus, brainstem, and spinal cord. The maximum changes are occurring in immobilization, stress, hypertension, and silica exposure. These are examples of neuronal damages, and you can see here that only hypertensive rats exposed to 33 neuronal changes are here, but together with the stress, they are much more worse in this situation. So in hypertensive rats as well, the neuronal damages are much more higher than the normotensive animals under similar conditions. Then comes how we can reduce these changes compounded by nanoparticles and hyperthermia. I can show you there could be many ways, but nowadays we know that many substances are released in the brain, and no single compound is responsible for all these things. We just given pre-treatment in these animals, and you can see TNF-alpha antibodies, 30 minutes, they are doing good. But when we have combined with titanium nanowire, the results are much better. One of the regions that how they are working here is upregulation of neuronal nitric oxide synthase, and you can see that titanium nanowire induced these antibodies have much better results. Here it's very clear. Now I can show you some example briefly that even diabetes can exacerbate brain damage. Here is diabetes plus silica, silica plus saline, and diabetes only in saline in two different brain areas. No stress at all. We have seen much more damage. These neuronal damages can be seen here. They are only diabetic animals exposed to silica dust, and you can see the changes were much more worse than silica and saline as compared to the saline treatment. So my point is that nanoparticles exposure on the one hand exacerbate brain damage, and then on the other hand, drugs delivered through nanotechnology can have much better effect. I will show you briefly some example. Here at electron microscopy, you can see that diabetes and silica has much more damage. The question occurs that whether size and age could be important. I briefly go to you some results here. This was presented at Society for Neuroscience. And so we have three range of the nanoparticles of three different materials: copper, silver, and aluminium. 20 to 3 nanometer, 50 to 60 nanometer, and 130 to 150 nanometer. We have three different ages: 9 to 10, young adult. This is the middle, 18 to 20 weeks, and 30 to 35, little older. And we have measured blood-brain barrier breakdown, brain edema, neural injury, and nitric oxide synthase expression. Here is the point. These are the very young, these are middle, and these are a little older rats. You can see that 20 to 30 nanometer size of copper has the maximum effect, whereas the larger size have less effect. So the size of nanoparticles are very important, so as the constituent of these nanoparticles. The constituent of nanoparticles is also important. It's shown here. These are the young, old, and middle age. You can see that copper, but the maximum effect we can see by silver, and lesser is aluminium. So even three types of nanoparticles with three different properties. Silver has the most toxicity, but the young and old are more vulnerable. The same thing is happening to the blood-brain barrier breakdown. The blood-brain barrier leakage is much more higher by silver and the small nanoparticles as compared to the larger size of the same nanoparticles. Cortical edema is also showing the same pattern, so as the volume swelling. Neuronal nitric oxide synthase is also quite parallel to what we have observed earlier, and neuronal damage is also showing similar changes. So the point is that each nanoparticle of same size also depends on its constituent. Silver is more damaged, and the small sizes, 20 to 30 nanometer, is much more damaging as compared to the higher sizes. This is showing a correlation, tight correlation. So you can see that silver and copper are more neurotoxic. Then we went to electron microscopy and immunohistochemistry. Here also, young, old, and adult, nitric oxide synthase expression is much more higher here, so as glial activation and so as neuronal damages. At electron microscopy, we tested our hypothesis, and you can see here that myelin damage is also similar. Young and old were much more vulnerable. These are Luxor fast blue, but some changes occur. The point is that adult animals can withstand up to some extent, but young and old were more vulnerable. So the neurotoxicity of nanoparticle size is very important, so at age. Now consider the situation in war. Our soldiers get injured, spinal cord and brain. I will only briefly tell you about spinal cord injury. I can tell that no model is good in experimental cases, because whatever we do in experiments, we are performing on healthy animals, and before performing any injury, we give them anesthesia. But in normal situations, patients or soldiers are injured when they are conscious. So we cannot replicate, but we have a model of head injury and also spinal cord injury. Here is the concussive brain injury. We drop a weight over the parietal skull, and it produces concussive head injury. We have another model about opening of the skull and making injury. Then the question comes, which drug we should try to deliver? I tell you, after injury or any condition, there are hundreds of thousands of chemicals are released in the brain. So if we want to block only one chemical, nowadays it is not possible to cure everything that we have. So we should have a multi-modal drug that can have different properties and working at different angles. At the moment, we are concentrating on Cerebrolysin. This is an Austrian drug, and this is a multi-modal drug. They have different types of actions. It helps in neuronal survival, neuroprotection, neuroplasticity, and even neurogenesis. Because this is a balanced composition of several neurotrophic factors and active peptide fragments. We have a couple of investigations done, and we found very good results. So we are working on this drug alone and also given as a nano drug delivery. Here is Dr. Ryan Tian at University of Fayetteville, Arkansas, and here is Dr. Wang. They are the discoverer of titanium nanowires. They have the US patent, and we are working with them to work on nano drug delivery. This is Dr. Yang. He is really the discoverer of nanowires. And you can see that nanowires help even stem cells to live longer and can have better effect. This is an example when they were placed on nanowires, they can live longer and produce better results. This is an example of our titanium nanowires. And earlier, this nanowire technology of delivery of Cerebrolysin got top 20% technology award at National Innovation Summit in the United States in Washington, D.C. in 2013. So we are much more encouraged. And also, our results came on the spotlight. Well, we have several sessions arranged on Nano Symposium in Society for Neuroscience. But look here, in concussive brain injuries, we have seen 24-hour and 12-hour. I'm showing some results. The left half is much more damaged when the injury is done on the right side because this is concussion. And here, when we have given Cerebrolysin in different doses or with nano delivery, we can see that in nano delivery, the small doses are much more effective. But when there is no nano delivery, we have to increase the dose to have the same effect. And also, this is occurring with glial cells. They are much more excited here. And at the electron microscopy, when the nano delivery of Cerebrolysin was given, even after 24 hours, we can see that there is much more better neuroprotection when we give normal Cerebrolysin. Here is the example of neuropay. We have much more better results with nano delivery of Cerebrolysin. So we can say that Cerebrolysin, when given with nanoparticles, even after 8 hours of injury, is doing much better. And the other factors are working even cold and hot environment. I do not have time to go with that. And the problem in clinical cases that why we fail, because no two clinical cases are very similar. They have many other factors. So therefore, we must consider many other aspects before devising any clinical treatment. I will go briefly with this nano delivery of Cerebrolysin and Alzheimer's disease, only 2 minutes. So, Alzheimer's disease here, an example of histopathological senile plaques. And what we have done, we have a model, then we infuse amyloid beta peptide for 4 weeks intracerebroventricularly to produce Alzheimer-like effect in rats and also in mice. So what you can see that A beta level is increased in the infused animals. Cerebrolysin alone given has reduced it, but nanowire Cerebrolysin has the best effect. The same case with happening with tau protein. In infusion alone, it has increased severely. Normal Cerebrolysin and nanowire Cerebrolysin has really came to normal control level. This is Dr. Shelley. We are working now with him, and he would like to see small fragments of CRF on which he got the Nobel Prize that we nanowire and infuse to see on neuroprotection. These results are coming. Also, this Dr. Shikanova I showed, and this is Ole Petter Otterson, who was the chief editor of neuroscience. Now he is rector of Oslo University. He also supports our results, and we are working with him. And since nanoparticle research are very expensive, we have the support of past governor of Arkansas and even the current governor of Arkansas. In the spinal cord injury, we also see the same effect. Here is our model of a spinal cord injury, and silica exposed spinal cord is much more expanded in damage as compared to the normal. And here you can see that silica exposed spinal cord much more tissue injury than the normal saline treated. And here is the current Arkansas governor, Mike Beebe, who is also supporting our nanoparticle research. And we have analyzed our results impartially, and you can see that in this part, when the drugs are there, they are the most innocuous because untreated is lying here. Some of the drugs, nanowired here, they are here, but some of the drugs are in between. So my point is that only nanowiring the drugs will not help you. You have to have good drugs that should be given with nanowires to have the potentiate their effects. That is very important. And this is an example. We have a drug nanowired AP713. When given in a spinal cord injury, it is much more better than the untreated one. And also, our former Prime Minister of Sweden is actively engaged in these research because these are so expensive. Now we are working with the government of China. This is Bethune International Peace Hospital. Is our friend Dr. Lian Yuan Feng. And we have lots of supporters in this area. Several organizations fund us. And these are our supporters in different areas. Ryan Tian from the United States, John Schlager from United States Air Force Research Laboratory. And I stop here to have your feedback. Thank you very much for your kind attention.
Thank you very much, Professor Sharma. It was absolutely interesting and very originally presented lecture. За мен това беше една изключително интересна и оригинално представена лекция, която хвърли светлина както върху невротоксичността на използваните наночастици в наномедицината. От една страна, от друга страна пък бяха представени възможности, които ни предоставя наномедицината за лечението на някои сериозни неврологични състояния, които са гръбначна мозъчната травма. Ако имате някакви въпроси и коментари, заповядайте, колеги. Ако няма, ще си позволя да направя един кратък коментар. В исторически план, може би са изминали над 60 години, откакто за първи път беше въведено понятието нанотехнология от един американски физик, Пайнан. Но още по-интересен дата е 1986 година, когато е издадена книгата на Ерик Дрексер, който се смята всъщност за гуру в наномедицината и всъщност той е пророка на нанотехнологиите. И в този смисъл аз бих запитал професор Шарма, вижда ли възможности за достигане на това, към което се стреми човечеството като безсмъртие или удължаване най-малкото на човешкия живот, като използване възможности на нанотехнологията и на наномедицината, особено когато става въпрос за нелечими до този момент заболявания? I'm going to translate my question. Professor Sharma, in the context of your presentation, I would like to ask you, what is your opinion about the immortality or prolongation of lifespan concerning the use of methods of nanotechnology in the nanomedicine?
Okay. The question is that what is the use of nanoparticles in nanomedicine and longevity of animals, I guess. So I can tell you that the metal nanoparticles, depending on their size and type, they can produce damage, whereas there are some other metals, they are very innocuous. As I have shown that aluminium is much less toxic. We are also testing this titanium nanowires, they are much less toxic. But on the other hand, silver is very toxic, followed by copper. So you have to choose